Hello dear friends, Namaskar, how are you? I first of all congratulate all of you for having successfully completing the BSc first year course last year. Now, I welcome you in the BSc second year class in the subject of Zoology. Today, we will discuss about the genetics part 2 in this semester of this year. Now, this is the third semester that you are going to study, and whatever you have studied in last semester, that is the genetics part first. In the genetics part first, we have already discussed various things about the genes, Mendelian laws of inheritance, gene interactions having the supplementary gene, complementary genes, different methods of sex determinations, and many other basic things. Now, this is the continuation of the genetics part 2. Previously, in first semester, you have already seen about the cell biology, that is the structure of cell, the structure of nucleus, structure of chromosomes. So, I think you have sufficient knowledge about the basics of the structure of cell and the structure of the genes, etc. But now, for continuation of again here in this paper, we are actually going into details about the studying the gene expression, what is the genetic engineering, microbial genetics, human genetics, etc. So, first of all, start today in this lecture we will going to see what is the gene, structure, concept, definition, function, etc. Friends, as you know, the genes are studied in the genetics, and the genetics is the branch of biology which actually deals with the study of hereditary material, ultimately the study of genes. Friends, the genes are as important as like the atoms in chemistry. The genes plays a variety of roles in the body of every living organisms, and therefore genetics is again becomes an important branch of biology in which all these situations and the structure and function and role of the genes and related to the genes have been studied. So, again the genetics is the branch which is associated to or applied for the agricultural area, medicinal biology as well as the biotechnology. So, the genetics affects the all the applied mechanics present in our life or which are very needful in our life. Nowadays, you know that we are facing one of the most serious pandemic disease called as corona, and all time, everywhere, every nation is going on by discovering the medicine over this coronavirus. So, again they are studying the patterns of the genes and the gene interaction present in that particular virus. So, what are genes? First, you again have definitely understood that the genetics is the study of heredity in general and genes in particular. So, the main focus in the genetics is the study of genes. Now, what is heredity? The heredity and genes are not apart from each other. So, first of all, we will see what are the genes. Friends, as you know, the genes are very minute, teeny structures located on the chromosomes, and they are important structural and functional units of heredity. Now, friends, as I told you, the heredity is the transmission of the parental characters to their offsprings. So, hereditary genes are present which actually helps for transmission of the particular character from one generation to the next generation, or from parent to their offsprings. So, this is about the hereditary characters. Friends, there are numerous characters which are remain hereditary and linked with the genes which are transmitted from one generation to other generation. We must heard about many things, that this son looks like a father or his grandfather or his grandmother, etc. So, all these things are related with the study of the face, with the related study of the eyes, with the related study of the hairs and the body pattern, etc. So we are trying to matching a particular child to his someone elder ones or to their previous generations. Now what it tells us that the particular child or particular offspring has some relations to their parents. So this relationship is brought about to himself by the particular genes. So genes are very, very important one. The genes can be defined in many ways, like they are the basic physical and functional units of heredity, or genes are important again which make molecule for protein synthesis in the living organism. Friends, again there are alternative forms of genes which are called as alleles. So when genes are present in alternative forms, they are called as respective alleles of each other. Coming towards the history of gene. Friends, as you know, the evolution of the microscope, the first microscope have been discovered in 17th century by Robert Hooke, whereas later on the evolution, as the evolution progresses in the microscope, the development and evolution in the study of the cell biology as well as in the genetical science has been evolved. During 19th century, the study of the gene has been expressed in more in detail because of the development of some of the advanced equipments in that particular century. The study or the invention starts in the 19th century. In 1909, a Danish botanist scientist named Johansen coined the word gene. But before that, in 1905, actually the Batson puts a word called as genetics. So this is the starting journey of the building up of the genetics or the study of genes in that particular era. Later on, in 1908, Garrod actually proposed a one gene, one product hypothesis. In 1911, T.H. Morgan have studied the gene theory, and later on, coming towards the half part of the 19th century, in 1948 and 1949, Pauling and Ingram actually established the role of genes in the protein synthesis, as well as the Beadle and Tatum proposed a one gene, one hypothesis theory. So again, in 1962, Benzere proposed a fine structure of gene. So this is actually the year in which the fine structure of the gene has been focused or localized or expressed by the Benzere. Later on, the gene concept was again elaborated by Sutton, and the work is going on for the decoding of the structure of the genes up till now. Friends, so with this we can say that until 1940 the gene was under established and was unexpressed, but or it was considered as a basic unit of genetic information. The role of the genes is limitedly studied during that particular period, but nowadays we know that the gene can mutate, gene can recombinate, gene can duplicate, or gene shows crossing over, and also genes are capable of synthesizing polypeptide chains of an enzyme, and their role in protein synthesis has been greatly expressed or known. So gene performs various types of roles we later on discussed in our next slide. So this is the another part of the gene which again we are having discussed in we are going to discuss in the next chapters. So basically the genes, the types of genes, there are so many types of or variety of genes which are performing their different role in our body or in any living organism. including microorganisms as well as in the higher organism also. So on the basis of behavior, the genes are classified into many types. One of the important type is basic gene. It is also called as fundamental gene because it actually brings about expression of a particular character. So it develops particular character in that specific individual. The another type of gene is lethal gene. Now these are one of the most dangerous genes which actually brings death of the possessor. So they are very harmful genes. If they occurs, the death may occurs in that particular possessor or wherever these genes have been located, they actually creates dangerous problem in that particular living organism. Example, cancerous cells actually we can call these are caused by the lethal genes. Then there are multiple genes. Now why they are called as multiple? Because they are again for producing single trait or single character, they need there are more than actually 2 or more genes are expressed to bring about that particular phenotypic character. The another type of gene is cumulative gene. Now these genes are shows additional effects on the action of the other genes. So they are supportive or shows their additional effects. Therefore they are called as cumulative genes. The another type is pleiotropic gene. The gene produce changes more than one character. So the genes brings about the different changes for producing more than one character. Likewise, friends, there are inhibitory genes. Now these inhibitory genes have particular role to inhibit the expression of the another gene, or they suppress the expression of the another gene, or they do not allow to express the another gene to show their particular effects. Now there is another type of gene which is called as modified gene. Now this modifying gene or modifier gene actually helps to modify the effect of or helps to modify the another gene, and likewise their effect has been modified. Besides these, there are as you know, we have studied in last year, there may be epistatic gene, hypostatic gene, complementary gene, dominant gene, supplementary gene, recessive gene, etc. Friends, on the basis of the gene action, the genes are divided into structural gene, operational gene and regulatory gene. So these are the general types of the genes which are located in the different parts of which are present in the living organism. Now coming towards the main part, that is the structure of gene. Friends, as you know, the genes are located in the chromosome, and the chromosomes are present in the nucleus of any cell. So these are the compulsory part which are present in any living body parts or in any living organism. Now these genes actually are the genetic information which controls the specific aspect of the phenotype. So what is phenotype? You know very well. We have studied the genotypic expressions and the phenotypic expressions. So it controls, the gene actually controls the phenotypic aspects. Now genes are made up of the deoxyribonucleic acid, that is the DNA. They are the fractions or part of DNA. They are not actual whole DNA, but they are a very small part or very small portion of DNA is called as the genes. Likewise, as DNA are made up of the adenine, cytosine, guanine, thiamine. Now these are the chemicals with which the genes can be made up of, or they are constituting the same elements within their structure. In most of the organisms, the DNA forms the main particular structure of the genes, and they are present in the sequencing type. Benzere in 1957 actually describe or divide this gene into 3 parts: cistron, muton and recon. So these are the 3 parts, namely cistron, muton and recon. So coming towards the actually position of the genes, as you know that they are arranged in a single linear order like beads on the specific loci of the chromosomes. So they are situated on the chromosomes and arranged like beads on that linear fashion. They are arranged on separate loci of the different chromosomes. Now friends, it is very important that if the position of the gene changes, the particular character or particular trait has been also shows their effect, or they also changes. The single gene, the single chromosome consists of many hundred to thousands of the genes, whereas the human genome actually consists of 20,000 up or to 25 or up to the 30,000 genes. The human genes are vary in size from few hundred bases to over 2 million bases. So the human gene actually consists of over 2 million bases. So such types of bases which are made up of the nucleotides, they have been, when counted, the maximum up to 2 million bases have been found in a single gene. So this diagram actually gives the entire picture about the structure of the gene. So this rounded part is of the cell. Within the cell there is a central nucleus, and from the nucleus a thread-like chromosomes. When we expand the thread-like chromosomes, we will see the double helix structure of DNA, and the small part of the DNA which actually the genes are located, or where the genes are present, which actually encodes or particularly helps in the protein synthesis. So these are the three diagrams actually which shows the ultra structure or ultra focusing images of the genes present in the cell or on the chromosomes. Friends, there are two types of genes, eukaryotic gene and prokaryotic gene, respectively present in the eukaryotic and the prokaryotic cells. Now what are difference we will actually study it in later lecture, but the eukaryotic genes are the important genes which actually shows or defined it into exon intron pattern, which having the promoter part enhancing portion as well as the termination portion, which actually encodes for the polypeptide chains for the synthesis of the polypeptides. Now coming towards the prokaryotic gene, this is very simple gene actually which shows this 5' N into 3' N, where actually these parts of the coding region and the regulatory region and the termination region has been observed and shows the RNA transcription. Coming towards the function of the gene, friends, as we have seen that the genes are very important one. As the atom is important in the chemistry, the genes are so much having importance in the genetics or in the biology. So now why they are so important? Because the genes actually is the unit of genetic information that controls the specific aspect of phenotype and specifies the synthesis of one polypeptide or protein synthesis. Again, you know that it transmits the character from one generation to another generation and called as hereditary material. So it helps in the transmission of the hereditary material. Gene contains specific information which actually necessary for the living cell to survive and reproduce. So they produce message, or they controls these actions for the living cells, for the survivability and for the reproduction. Genes again gives instruction to make a molecule called as proteins. However, many genes or some of the few genes also do not code for the proteins, but they have another role. Rather, from the synthesis of the protein, they play another role in the body. Now, genes control the functions of DNA and RNA. So we can say that the genes are generally associated with the production of enzyme, which they synthesize from chemical substance available in the body cells through the process of autocatalysis. Finally, again it is very important that one gene affects one enzyme. So this is the particularly needed things have been always observed, that one gene affects on one enzyme action. We can summarize the action or roles of genes likewise, that the genes have a very greater role in the development of pigments, in the development of colors, in the development of hormones, in the development of size and form of the individual, in the production of the proteins, in the production of antigen and antibodies, and they may also cause the severe diseases in the human beings like albinism and tyrosinosis and etc. So, this is the introduction of the genes and gene structure and the role of the genes in the particular living organism. So, we will stop here. Thank you.